Hi, my name is Lloyd Tran. I'm the Chairman and Chief Scientific Officer of Biomed Industries, Inc. in San Jose, California. I appreciate this opportunity to present to you a talk on the topic of reducing neurogenesis hypothesis using our clinical data of the drug NA-831 to illustrate the viability of the hypothesis for the treatment of Alzheimer's disease. Let me show you some slides. First, I'll introduce myself. I'm a scientist by training, have spent the last 20 years work on the field of dementia and Alzheimer's disease. I remember I filed my first patent application in the year 2001 on the topic of neurogenesis of the new drug for treatment of Alzheimer's disease. And my last patent, which was issued and granted by the US Patent Office, was on August 17, the year 2021. So 20 years working in this field. So I'm pleased to introduce you. Biomed Industries is a privately held clinical stage biotechnology company. We are committed to the development and commercialization of the new drug for the treatment of Alzheimer's disease. As you know, Alzheimer's disease is a progressive disease of the brain. It is a dreadful disease characterized by the impairment of the memory and cognitive functions, eventually provide and give rise to disturbance in reasoning, planning, language, and perception. In the US, there are 38 million people suffering from early symptoms of dementia. We call it MCI, mild cognitive impairment. Among them, about 8.5 million people have MCI due to Alzheimer's disease. And among these, there are about 5.7 million people in the US with mild and moderate Alzheimer's disease. In the world, there are about 35 to 40 million people worldwide suffering from this dreadful disease. The challenge is that there is a serious observation that almost all drugs of the past 12 years or so failed in phase 2 and phase 3 clinical trials. The list is so long, and listed on this chart. I like to highlight the fact that the reason why all drugs candidates fail in Alzheimer's disease because they based on the principle of amyloid hypothesis. This hypothesis believes that the disease, Alzheimer's disease, is caused by the buildup of the amyloid beta plaques. So a simple way to treat the patient is to find the drug to remove or to reduce the amount of amyloid on the brain. It doesn't work for the past many years. In fact, the amyloid hypothesis can be considered a dogma. A dogma is a belief or the set of belief that is accepted by the number of people in the group without being questioned or doubted. As you know, Biogen drug aducanumab has been approved by the FDA on June 2021. This drug did not show or doesn't show any kind of improvement in cognitive functions. In fact, more than 42% of the subjects participating in the phase 2/3 trials have amyloid-related imaging abnormalities, which is a brain swelling, and some of them cause fatal condition to some people. The FDA approved the drug aducanumab on June 2021 based on a surrogate endpoint biomarker, which is demonstrated by reduction of amyloid plaque, even though there was also any kind of improvement for the patient care or provide any kind of benefit for cognitive functions. Recently, the Centers for Medicare and Medicaid Services, known as Medicare, ruled on April 6, 2022, that to date, no trial of an anti-amyloid monoclonal antibodies has confidently demonstrated a clinical meaningful improvement in health outcomes, particularly cognitive functions of the Alzheimer's patients. Therefore, there is insufficient evidence to conclude that the use of monoclonal antibodies directly against amyloid is reasonable and necessary for the treatment of Alzheimer's disease. Biomed has introduced a new hypothesis known as the Neurogenesis Hypothesis as a potential replacement for the amyloid hypothesis. We present our hypothesis at the last conference of the Alzheimer's disease, Parkinson's disease, March 15 and 20th, the year 2022 in Spain. And we also present at the International Conference of Controversies in Neurology last March 24 to 27, 2022. So today, let me highlight the essential of neurogenesis hypothesis for Alzheimer's disease. The hippocampus is critical for learning and memory, containing neural progress. The hippocampus continues to generate new neurons throughout life. The process is known as adult hippocampal neurogenesis, AHN. Hippocampal neurogenesis is persistent throughout the 10th decade of human lives, up to 100 years of age, and is detectable in patients with Alzheimer's disease. Nestin substitute neuroprogenitor cells and DCX neuroblast and immature neurons were detected in subjects up to 90 years of age, according to publication by Moreno, Jimenez, and Tobin in the year 2019. There was a marked and progressive decline of DCX cell numbers in Alzheimer's patients as compared to healthy individuals. The neurogenesis framework, including neuroanatomy distribution of named neurons, it relates to the relationship between glial cells and apolipoprotein. The adult hippocampal neurogenesis impairment compromises the functions of the hippocampus in patients with AD and MCI. The adult hippocampal neurosis impairment appear to take place prior to the presence of synapses and neurofibrillary tangles, both of which are key pathological hallmark of Alzheimer's disease in the dentate gyrus. In other words, before the amyloid plaque can be detected in the brain, the process of neurogenesis impairment has taken place in the hippocampus, particularly in the dentate gyrus. So rather than observing the development of amyloid, we should look at the core of the disease, which is take place at the hippocampus of the individuals. Therefore, we propose the hypothesis and a new approach for drug development. We believe that stimulating inherent adult hippocampal neurogenesis could serve as a therapeutic target for improving cognitive functions and promoting synaptic resilience. I'd like to share with you the development of our drug called NA-831. This drug is a small drug molecule. It easily crosses the blood-brain barrier with great bioavailability. It exhibits both neurogenesis and neuroprotection in patients. We have finished the phase 1 and phase 2A. Phase 1 showed that the no adverse effect were observed, and it was well tolerated up to 50 milligrams per day. The phase 1 indicate that the predictable pharmacokinetics with dose-dependent exposure, linearity, and low variability. The phase 2A confirmed the proof of safety and efficacy of the drug for Alzheimer's disease. We plan to launch a phase 3 by end of 2022. The drug is an oral formulation, recommend up to 10 milligram to 30 milligram per day for patients with the early onset of Alzheimer's disease. With that, I'd like to tell you that NA-831 serves as a catalyst to restore neurogenesis in Alzheimer patients, increasing DCX and PCA NA cells with greater number of neuroblasts, leading to improved cognitive functions. According to Professor Nicolas Tony of Lausanne University Hospital in Switzerland, he said that even if drug companies could develop therapeutics that block or reduce the amyloid beta, they will not reverse the damage. But the adult neurogenesis in the hippocampus could be a way to partially right that wrong. In other words, the amyloid hypothesis does not help drug companies to find any new therapeutics because removing the amyloid does not help to improve the patient. But the neurogenesis makes a lot of sense because it helps to regenerate new neurons to offset the decline of the neurodegenerative process in the body. Over the past several months, we have presented our hypothesis as a number of conferences like the ADPD 2022 in Barcelona, Spain. We present at the World Congress of Controversies in Neurology, March 24 to 27, 2022. We also present the hypothesis at the New York Academy of Science on December 3rd, the year 2021. Let me summarize our phase 1 study. It is a safety, tolerability, pharmacokinetics, and pharmacodynamics of NA-31 in human volunteers. NA-31 was rapidly absorbed with Cmax, reached 3.28 microgram per millimole in plasma in about 1.75, so less than 2 hours. Post-dose with linear pharmacokinetics. PK, the mean serum half-life was approximately 6.5 hours, less than 7 hours. The coefficient of variance for both AUC and serum half-life was less than 20%. NA-31 was well tolerated up to the maximum dose of 50 milligrams per day in our phase 1 study. Conclusion, no adverse effects were observed. NA-831 demonstrates predictable pharmacokinetics, including dose-dependent exposure, linearity, and low variability. After phase 1, we proceed to finish the phase 2A study. It is a randomized placebo-controlled double-blind clinical trial of NA-31 performed a total of 56 patients, among them 30 patients with MCI and 24 patients with mild and moderate Alzheimer's disease. Patients with MCI received 10 milligrams of the drug or placebo orally per day, and patients with mild and moderate Alzheimer's disease received 30 milligrams of NA-31 or placebo orally per day. The key inclusion is that subjects with MCI to meet the NIA-AA core clinical criteria for mild cognitive impairment due to Alzheimer's disease. Subjects who have a CDR score of 0.5 and memory box score of 0.5 or greater at screening and baseline with MMSE score equal bigger than 22. Conclusion for subjects with mild and moderate Alzheimer's disease to meet with the NA-831 core clinical criteria is that the MMSE between the range 17 to 21. The primary outcome measures would change in Alzheimer's disease assessment scale according to subscale ADAS-Score 13, brief quality rating scale BCRS, and clinician interview-based impression of change plus caregiver input CIBIC plus after 24 weeks. The phase result can be summarized as follows. We found that patients on the drug treatment show significant improvement over 24 weeks in the following area: ability to concentrate, long-term memory, long-term and short-term memory, ability to self-care. Many Alzheimer patients, they lost ability to take care of themselves. Even they don't know where they are at home and they don't know where to find the bathroom in their own home. So they lose the sense of orientation. So the drug helped them to restore the ability, the sense of orientation, ability to self-care, and also the most important part is it improved the cognitive functions, which can be demonstrated using ADAS-Score 13. I'd like to show you this graph here. It is a ADAS-Score 13 scale change for patients with the MCI as compared with placebo. In the graph, the blue line at the bottom deteriorated, slowly declined, as compared with the red line represent the cohort of patients take the drug, and the difference between the red line and blue line after 24 weeks, the unit about 3.4. For the cohort of patients with mild and moderate Alzheimer's disease, the ADAS-Score 13 scale change is more profound. You see the blue line represent the placebo, the red line represent the patients taking the drug, and the difference is about 4.1. In summary, the NA-831 show a significant improvement of for MCI patients with the ADAS-Score 13 score of an average of 3.4 as compared to the placebo after 24 weeks of treatment, p equals 0.01 ITT. NA-31 show a significant improvement for patients with mild and moderate Alzheimer's disease with the ADAS-Score 13 score change of an average of 4.1 as compared to placebo after 24 weeks of treatment, p equal 0.001 ITT. CIBIC plus shows 78% patient improvement with p equal 0.01 ITT. NA-31 was well tolerated at 30 milligrams per day amongst the cohort of mild and moderate Alzheimer's disease. For people in this group report minor headaches and diarrhea, there was no serious adverse effect observed in both MCI and patients with mild and moderate Alzheimer's disease. In conclusion, we have demonstrated proof of safety and efficacy of NA-31 as a potential disease-modifying drug for improving cognitive functions and overall quality of life for patients with MCI and patients with mild and moderate Alzheimer's disease. We plan to begin enrollment process to do 2 programs of phase 2B and phase 3. One program is for the treatment of NA-31 in 475 patients with mild and moderate Alzheimer's disease over 52 weeks. The second program is for the prevention. We're to study the application of NA-31 in 585 patients with no symptoms because asymptomatic patients over 2 to 4 years old. We like to compare our drug with all the drugs receive a lot of attention in the market, particularly Biogen's aducanumab, Aduhelm. As you know, Aduhelm approved by the FDA, but has not widely used, and the Medicare have put the drug on hold, required the company have to conduct another phase 3 studies on about 1,500 patients in which Medicare will reimburse. Besides that, Medicare would not reimburse the drug, even though the drug has been approved by the FDA. Biogen drug costs about $28,000 per annum per patient, whereas our drug costs about $12,000 per year. I'd like to point out that aducanumab, Biogen drugs, is a monoclonal antibodies requiring intravenous administration once a month over the first 8 months using expensive PET scan or MRI, and in addition to a lumbar puncture, which is a very painful procedure for many patients. The cost of Aduhelm is $28,000 for the drug per year, plus about $40,000+ for PET scan, lumbar puncture, to total about $75,000 to $80,000 per year. Whereas our drug, NA-831, costs about $12,000 per patient per year, and the drug doesn't require any kind of brain scan, PET scan, MRI, neither lumbar puncture is required. So it's similar. It can be used by a lot of people in the US, worldwide, orally, and was safe and very effective for treatment, also for prevention. Intellectual property, Biomed Industries is the exclusive owner of all patents and patent pendings in the US and worldwide. The company holds all market rights of our intellectual property worldwide. The issue patent and patent application cover the drug NA-831 and its analogs for the treatment and prevention of mild cognitive impairment, Alzheimer disease, neurodegenerative disease, depression, and psychological disorders. As mentioned earlier, our latest US patent number 11,090,3.3 was issued on August 17, 2021, with the expiry date given protection up to May 2039. Besides the patent protection, we also rely on trade secrets and know-how to develop, to protect, and to maintain our competitive advantage. In terms of strategic planning, the company is pursuing a new track strategies. We are planning to do the IPO. We have signed the agreement with the underwriter to raise $50 to $75 million to fund our phase 2B and our 3 clinical trials for NA-831. We have entered exclusive marketing with a market pharmaceutical company in Australia and New Zealand. The agreement provides upfront payment and royalty of sales for NA-831 and our delivery system, Micros, in New Zealand and Australia. Besides that, we are in discussion with major US-based pharmaceutical companies for potential M&A. We're also in discussion with some SPACs with possibility for a potential business combination with a public company. With that, thank you very much for your interest and attention.